When you think of a genetically modified organism, you probably think of some crazy scientist in a high-tech lab genetically engineering a tomato that's barely edible. You might even use it as an excuse to buy all organic produce at the grocery store. But did you know that humans have been genetically modifying plants and animals basically since we started farming thousands of years ago? The ancient history of GMOs, up next on Fact Bites. And before we dig into it, don't forget to like, subscribe, and hit that bell for notifications. At its basic definition, a GMO is any plant or animal that's had its original genes, that's DNA and RNA, altered in some way. Today, we can do that in a science lab, but genetic modification can happen with much less sophisticated equipment. In fact, there are foods today that we wouldn't even have without some form of modification. And not just the processed and packaged stuff either, but basic staples of our global diet. Take corn, for instance.
Paleobotany has pinpointed corn's domestication to about 9,000 years ago. Corn, or more commonly known as maize, was domesticated in what is now modern-day Mexico and South America. But prehistoric maize looked very different from the corn we eat today. Corn is actually descended from a grass called teosinte. If you look at the top of a stalk of teosinte, you can kind of see how we got from this to corn. There is a single string of kernels that almost resemble a corn cob, but it's a single strand with a tough exterior and a not-so-great taste. So how do we go from this tiny strand of kernels to the juicy ears of corn we eat now? Selective breeding. By collecting kernels from teosinte that had the softest casings, most abundant stalks, and the sweetest taste, the ancient peoples of Mexico were able to grow plants that were bigger and more palatable than their teosinte forefathers. This is where we get maize from.
As maize became a staple food source in the culture, they began trading it with other groups, and it eventually made its way to the Amazon region. Now, maize was, and continues to be, an easy crop to farm, so it took to the soil in South America as easily as it did in Mexico. The big difference, though, was that in South America, the maize plants were no longer cross-pollinating with the teosinte grasses.
Because these two plants were so similar, they were able to cross-pollinate and fertilize each other, so the maize grown in Mexico stayed smaller and less appetizing for longer than its Amazonian offspring. In South America, however, there was less teosinte grass. This meant that the maize plants were only pollinating with each other, resulting in larger, sweeter, juicier ears of corn than in Mexico. This new species of maize continued to flourish and ultimately moved back to Mexico and then further into North America. Today, corn and corn byproducts like cornstarch and corn syrup are major staples in our everyday lives. In fact, corn makes up about 10% of our global crop production to feed humans and animals alike and support industries like ethanol and plastics production.
And none of that would have been possible without human intervention and the genetic modification of selective agriculture thousands of years ago. So corn was technically edible, just sort of unpleasant, but what about downright poisonous plants? Let's look at almonds.
Wild almonds contain a chemical compound called amygdalin. If a human eats a wild almond, that amygdalin breaks into two other chemicals: benzaldehyde, which makes a bitter taste in your mouth, and cyanide, which makes you dead. But a natural genetic mutation in some almond trees' alleles inhibited the production of amygdalin, making almonds sweet and safe to eat.
Okay, I gotta know, who was the human that discovered the sweet almond? Like, were they dared to eat a bunch of almonds and just happened to pick the one tree that wasn't poisonous? Were they just real invested in almond trees and told everyone, no guys, we can make this work, I swear?
Anyway, upon discovering the sweet almond trees, humans figured out that you can actually change the dominant allele that produces amygdalin by driving a piece of pine wood into the roots of the tree. It's believed that this stresses the tree out enough that it turns that allele off. The kicker? This all happened at least 3,000 years ago, although some geneticists believe humans were domesticating almonds as early as 12,000 years ago. That's earlier than corn. Let's take a moment to appreciate that humans were changing genetics before they even had words for genes, alleles, or knew about the chemical compound amygdalin. Both Pliny the Elder and St. Basil's Hexameron detail the method for turning bitter, poisonous almond trees into the delicious nuts we know today. Through that selective agricultural process, mankind was able to breed out the amygdalin, and most of the almond trees we have today are the non-poisonous kind. But there's still some out there, so friendly reminder to not eat just any old almonds you find out in the wild. It's theorized that humans were able to domesticate almonds so quickly because they were able to cultivate the trees from seeds.
Fast forward in time a little bit, and humans started using slightly more sophisticated methods for genetically modifying plants to produce bigger, better, tastier results. Let's look at the banana.
If you found a banana in the wild, it would look a lot different from the banana you find in the grocery store. For one thing, it would be almost entirely seeds and almost no fruit. For another, it probably wouldn't be as sweet as the bananas you're used to. About 5,000 years ago, farmers in Papua New Guinea were able to selectively breed bananas into a variety of seedless or near-seedless fruits. Although most grocery stores only carry that familiar yellow variety, you can still find some variants like plantains and red bananas in international markets. However, over the centuries, the big yellow variety dominated.
Here's the thing about bananas, guys. See, we cultivated bananas to be seedless, which creates a bit of an agricultural conundrum. How do you grow new banana plants if they don't have any seeds? Mankind figured out that they could take cuttings off the banana plant to propagate new plants. Essentially, you cut a piece off the first banana plant, put it in a new plot of dirt, and let it grow. Which is pretty ingenious when you think about it. It means that you're growing the exact same banana with the exact same traits. No seeds, easy to peel, sweet flavor, because you're basically cloning the banana. I mean, talk about product consistency. Except, if all your plants are clones of each other, that means that they're all susceptible to the same diseases. And if one plant picks up a disease, then all the plants will pick up that disease, which is exactly what happened to the banana in the early 1900s.
See, before 1950, the majority of bananas consumed around the world was the variety Gros Michel. I'd love to tell you what a Gros Michel tastes like, but unfortunately this strain of banana is almost extinct. Starting in Southeast Asia and then migrating to the banana plantations in Latin America, a fungus called Panama disease, or Fusarium wilt, started wiping out entire crops of bananas. As the global banana population dwindled, scientists and fruit companies started looking for a banana replacement. They settled on the Cavendish banana that was both similar in size, shape, and taste, and also resistant to the still pervasive Panama disease.
So the banana industry was saved! Or so we thought.
Growing in the wild, the Panama fungus started to mutate, and in the 1990s, it started affecting Cavendish plantations across Asia and Africa. Although it hasn't gotten a foothold in Latin America yet, the banana industry is concerned about another population wipeout.
So what will the banana of the future taste like? Who knows? But they better figure it out soon, because bananas are the fourth most valuable global food crop, right behind wheat, rice, and milk. So if bananas disappear, we're kind of in trouble. So see, humans have been impacting the genetics of plants for centuries, and this is just a handful. There's tons more. Peaches, tomatoes, squash, foods that make up our everyday life wouldn't exist without just a little bit of genetic manipulation from our ancestors.
Today, foods and plants labeled as GMO are more likely made in a lab where a geneticist is physically replacing strands of genes to make more abundant and resilient crops. This advanced manipulation makes for faster changes in genetic structure, unlike the thousands of years it took to cultivate the plants we've gone over in this episode.
Got any other foods that look different from their ancient predecessors? Let us know in the comments below. And don't forget to hit like, subscribe, and that bell for notifications so you're always ready for the next crop of Fact Bites. Thanks for watching!